Welcome back, friends. Welcome to another video session from Shomu's Biology. And in this video tutorial, we'll be talking about membrane fluidity. You probably heard this many times about this particular term, membrane fluidity. Now, what it means actually? Membrane fluidity means we're talking about the cell membrane fluidity. Now, cell membrane is composed of lipids that we know. That the cell membrane that we know of is mainly composed of lipid molecules. Those are known as phospholipids. Phospholipids. And phospholipid molecules have specific structures. They have one head-like structure which is hydrophilic, and it also have another structure which is known as the hydrophobic tail. Hydrophobic. This is the tail. This is the head. This is the structure of the unit of a cell membrane. Now, a lot of these phospholipid molecules, they are joined with each other and they form the cell membrane. And these phospholipids, they first join with each other by a single layer, like say all the heads together, all the tails together. This is a single layer or monolayer. Such we have another same layer, and here we go. So it is a double layer. Okay. Now both these layers are containing this phospholipids. Now if you look at here, this is the structure of membrane. Now if we zoom into here, this will form the cell membrane like this. Rest of the thing is filled with this membrane. Now if you look at this structure very closely, you will see this outer portion of the phospholipid head is going to interact the outer environment, while the inner part of the phospholipid head is going to interact with the inside of the cell, that is the cytosol. Now these tail regions in the middle, they form the hydrophobic core, which is not interacting with anyone or else here. Okay. Now the thing is, the mobility or the fluidity of membrane represents the movement of these phospholipid molecules inside in this cell membrane, because they move, they migrate from one place to another place. Because if we consider this cell membrane as a floating, everything here is like a floating ocean. I mean, everything is floating in the ocean, like some balls are floating in the oceans. So in that case, all those things are moving in a floating space. That is how you can consider the cell membrane structure actually. Now in this membrane, there are proteins also embedded. Maybe that protein can be attached someplace in the outer area like this. The proteins may be sometimes embedded through this cell membrane. That protein can be interact someplace inside and anchored to the cell membrane like that. Now the thing is, this movement of phospholipids are very necessary for so many purposes. Like example, these proteins will be placed from one place to another, right? Because maybe let's say some inner leaflet enzyme is present here. That enzyme sometimes needs to interact with protein, another protein that is present in the membrane. So that two proteins need to come close to each other for a proper intracellular signaling. For example, growth factor comes in from outside. When the growth factor is attached with the growth factor receptor, they require two of the membrane proteins to come closer so that the cell can grow. Now for all these biological activities, the cell needs to migrate its proteins as well as needs to migrate its lipids as well. Now this is the fluidity of the membrane, the dynamic nature of the membrane. There are two terms that we can say. One is membrane fluidity, another one is the lipid mobility. Now those things are very much closely related. As I told you, the membrane fluidity means the dynamic nature of the membrane, while the membrane mobility of the lipid means this closing, or you say this movement of lipids and movement of proteins in the cell membrane. This is the mobility, while the fluidity is just the dynamicity. And to maintain the dynamicity, the mobility or the migration of those lipids and proteins are very important. Now the question is, what kind of fluidity we observe and why we observe membrane fluidity at all. Now the idea is the membrane fluidity, as you know, is important thing because we need this membrane to be dynamic to properly function. Now that fluidity is controlled by three things, three major things that are controlling the membrane fluidity, or which is helping the membrane to remain as fluid. Now if I delete this part and let you know what the sequences are, you will know that. Let's say here you see if this is the membrane. Now the arrangement of these lipids and those head of the lipids and the tail of the lipids can vary for different types of cells and different situations in different temperatures and conditions. Now the idea is, let's say if we increase the temperature a little bit, if we change the temperature a little bit, what it does actually, it creates less, it creates the fluidity more. Why? Because those lipid molecules which are interacting with themselves, as the interaction was strong earlier, but as we increase the temperature, the interaction gets less, and as a result the fluidity increases with temperature. So what happens actually in this case, those lipids now will be distantly placed, not that much tightly placed like earlier, like this for example. So you see, in high temperature there is, the mobility is increased, the fluidity is increased, and there are two different forms of fluidity we can observe. The first form that we are looking at here, which is very compact form, that we can achieve at very low temperatures. When the temperature goes down, all those hydrophobic regions of the lipids tend to stick to each other more compactly, more tightly, giving a very compact structure, and that structure is known as the crystal structure of the cell membrane. On the other hand, if we increase the temperature, that crystal structure gets distorted, and as a result what we get in turn, we get much destabilized structure, less compact structure, and that is known as the plasma structure, or like solution or salt structure, or gel like structure you can say. So these are the difference, crystalline structure and gel like structures depending upon the change of temperature. Now imagine some animals then tends to stay at very cool climates, for example Arctic fox, for example polar bears. Now they are staying at very cold climates, so how would they survive in those situations? Now in that case it's very important, because in very cold conditions the cell membrane gets too much complex, too much condensed with each other, everything is compact. The movement of proteins and lipids are very difficult. Not only the movement of proteins in the membrane, but also let's say there are some molecules that needs to pass the membrane, that would be very tedious. So how could those organisms survive in those conditions? Now to answer that, we need to know what are the elements that are controlling this change in the membrane components, this change in the membrane integrity. And the answer for that is, as I told you, three things. So let me list three things here. First I will write is cholesterol. Okay, I put the example as temperature. As you know, temperature is a factor. I am not going to include that here, that's why I tell the integral factors which controls this transition, state transition and stuff. One is cholesterol, second one is the state or nature of the phospholipid, and the state of the phospholipid, that means whether it is saturated or unsaturated. Okay, that is the thing. And third is the length of those phospholipids. Okay, mainly these three things: cholesterol, presence of cholesterol in the membrane, the nature of those phospholipids that are present there, and the length of the phospholipids that are present. Now if we look at here the idea, first thing is let me first talk about the length of the phospholipid. It's all about the structure's compactness. Okay, now the idea is, if this region of hydrophobic region, which is known as the hydrophobic tail of the lipid, if this region is long enough or larger, long enough space, that will provide these lipids more room or more area to properly interact to the neighboring lipid, right? So just assume two scenarios. In one scenario, what we have, let's say this is the head and the tail, and this is another head, say this one, head with another tail, head with another tail. On the other hand, let's assume the scenario like this. So this is scenario number one that I want you to focus. Now in this scenario, you will see that this first one, they carries longer hydrophobic tail. That means they get more room to interact with themselves with the help of this long tail. On the other hand, in this case, they will not get more room to interact. So this interaction will be more complex, more compact and strong compared to this. This is one thing. So longer hydrophobic tail will help to organize the structure more. So it becomes much more compact. Second is that state or nature of the phospholipid. Now the phospholipids that we know, they can be of two different types, saturated and unsaturated. Now saturated and unsaturated, the difference means in case of saturated fatty acids or any kind of saturated fatty acid if we choose, in that case you will see, let me go and explain from this angle, that in saturated and non-saturated they have different types of kings, okay, like this. For example, this is the actual structure kind of for fatty acids. In some case they contains double bonds in those, like this. So if they carry these double bonds, that double bond creates kinks. Kink means the fatty acids without those double bonds, they behave some structure like this, but the fatty acid with double bonds would behave something like this. So they are much distorted. The structure is more distorted. Okay, it should not meet here, but it will form a kink and the structure will be distorted. Okay. Now in this case, this is unsaturated fatty acids. In unsaturated fatty acids, you will see that excess double bonds, and double bond means there will be a kink. Now those kink will make it much less organized. So if the concentration of lipid molecules contains most of or more of this unsaturated fatty acids, in that case what it will do, unsaturated fatty acid will make the compactness less, because if they are present in the middle, let's assume now if you place the same thing and you place the unsaturated fatty acid in the middle, then what will happen? Let's say here this is the unsaturated fatty acid, so it forms a kink like this, so the nearby organization will be gone. Okay, but if it contains more of this saturated one, in that case it will become more compact, the membrane structure will become more compact. That is the idea of saturated and unsaturated fatty acids. The third thing is the cholesterol, and this is the most important one among all, okay, and where people get more confused. The presence of cholesterol impacts the fluidity of membrane in so many order, okay, and it affects two different way. Now first of all, cholesterol is a molecule which also contains two different domains, like one hydrophobic, one hydrophilic. The hydrophilic region of the cholesterol is nothing but hydroxyl. Only hydroxyl group is acting as a hydrophilic group for cholesterol, and rest of them are hydrophobic. And this is a bulk structure of chain and rings that are present. They act as a hydrophobic region of the cholesterol. So the cholesterol molecule usually placed in the membrane by the fashion, as you know, that hydrophilic should interact with hydrophilic, hydrophobic should interact with hydrophobic. So if we see or place the hydrophobic cholesterol somewhere, let's say in this case, let's say here we'll place the cholesterol, then how it will look like? It will look something like this. Here it will be the hydroxyl group of the cholesterol. Let's say this is the hydroxyl group of the cholesterol, and rest of the bulkier rings that presents here. So cholesterol will interact somewhat like this. Okay. Now the question is how it regulates the structure. Okay. Now imagine two scenarios. The scenario where there is the membrane is undergoing high temperature sensitivity, and another thing where the membrane is in low or cold temperatures, low temperatures. So if a membrane is in cold temperature, just imagine this is a cool climate where all those phospholipids are tend to place with each other, very close attachments. In that conditions, if cholesterol is present, then cholesterol is going to hamper the tight interactions between the neighboring phospholipids. It will not allow those phospholipids to properly engage and attach with themselves because of its bulky ring. It will prevent them. So in cold conditions, in low temperatures, if more cholesterols are found in the membrane, those cholesterols will make the membrane more permeable and less complex, less compact, which is very important and very necessary. As I told you, there are animals which are living in cold conditions and cold climates who want to survive, and they can survive by this fashion with the presence of producing more cholesterol and adding them in the membrane, right? Now comes down to another scenario. Let's say now the temperature is elevated and now the cell membrane is under very high temperature, and very high temperature, we know that the phospholipids tends to separate from each other. So they are separated, as you see, they are separated. Now in this high temperature, if cholesterol is present in the membrane, then cholesterol is going to, let's say here is the cholesterol, let me draw it with red color. Let's say here is the cholesterol with this hydrophilic group and then the bulky ring. In that condition, as you know, cholesterol will try to interact with the, because this bulky ring of the cholesterol, that is the hydrophobic domain of the cholesterol, it will try to interact with the nearby hydrophobic regions of the lipid. But here lipids are distantly placed. So what cholesterol want to do? If I am a cholesterol and I want to engage interactions, hydrophobic interactions, I see things are in the distant place, cholesterol will try to grab those hydrophobic tails. So he'll try to grab them and put them close to each other. So at this very high temperature where you see the membrane is more permeable and the structure is very disorganized, and that condition cholesterol can change the organization, and cholesterol will bring all those phospholipids close to each other so that their integrity comes back to the cell membrane. So cholesterol functions both way. When the membrane is too much compact in the crystalline state, presence of cholesterol at those low temperature conditions will make the membrane more permeable and more fluid, while at very high temperature, when the membrane is too much fluid, presence of cholesterol will make the membrane more complex and compact and less fluid. The reason behind it is the structure of cholesterol and how cholesterol interacts and engage itself with the nearby lipids. That is how presence of cholesterol controls the movement or change in the fluidity of membrane from the crystalline state to the liquid state, and again from the liquid to the crystalline state. It's very, very important. So for all those organisms who are doing this thermoregulation processes, in those organisms, they always rely on these three things, like they produce more unsaturated. If the organism is placed in a very cool climate, very cold climate, in that conditions to survive, for example Arctic fox, as I told you, what they will do, they will produce more unsaturated fatty acids, so that those unsaturated fatty acids make less interactions with each other and creates more fluidity, and they will also produce more cholesterol, so that cholesterol will further increase the fluidity. Okay. And if you look at the high temperature organisms, in that case they will do the opposite thing, right? So this is the way organisms control membrane fluidity, and this is why membrane fluidity is very, very important thing to consider. So that in a sense is a membrane fluidity, and I hope you learn what is membrane fluidity and the role of cholesterol in membrane fluidity. Thank you for watching. If you like the video, please hit the like button, share this video with your friends. Thank you.